Polymerase chain reaction, PCR. PCR, polymerase chain reaction, is a laboratory technique used to make millions to billions of copies of a specific DNA sequence in vitro, outside the cell. It mimics natural DNA replication but is carried out in a test tube using a thermal cycler. Invented in 1983 by Kary B. Mullis, who received the 1993 Nobel Prize in Chemistry for this discovery. Discovery of PCR. Kary Mullis developed PCR while working at Cetus Corporation in 1983. The key breakthrough was the use of a heat-stable DNA polymerase from Thermus aquaticus bacteria, found in hot springs. This enzyme, TAC polymerase, could withstand high temperatures used for denaturation, eliminating the need to add new polymerase after each cycle. Components of PCR. PCR requires a few simple but essential components. Template DNA, the DNA that contains the target sequence to be amplified. Primers, short single-stranded DNA sequences, 18 to 25 bases, that flank the target DNA region. They provide starting points for DNA synthesis. DNA polymerase, usually TAC polymerase, heat-stable enzyme from Thermus aquaticus. DNTPS, deoxynucleotide triphosphates, building blocks, DATP, DTTP, DCTP, DGTP, used to synthesize new DNA strands. Buffer solution, maintains the optimal pH and ionic conditions for enzyme activity. Mg superscript 2 ions, essential cofactor for DNA polymerase activity. Steps of PCR. PCR consists of repeated cycles, 20 to 40, of three main steps inside a thermal cycler. Denaturation, 95 degrees Celsius. Double-stranded DNA melts into single strands by breaking hydrogen bonds between base pairs. Annealing, 50 to 65 degrees Celsius. Primers bind, anneal, to their complementary sequences on the single-stranded DNA template. Temperature depends on primer sequence. Extension/elongation, 72 degrees Celsius. TAC polymerase synthesizes new DNA strands by adding DNTPS to the primer, complementary to the template strand. Each cycle doubles the DNA amount, leading to exponential amplification. Applications of PCR. PCR revolutionized molecular biology and biotechnology. Major applications include: 1. Medical applications. Disease diagnosis, detecting pathogens, example HIV, tuberculosis, SARS-CoV-2, COVID-19 testing. Cancer research, detecting mutations in oncogenes/tumor suppressor genes. Prenatal diagnosis, identifying genetic disorders, example cystic fibrosis, sickle cell anemia. 2. Forensic science. DNA fingerprinting, rightward arrow, identifying criminals, victims, or paternity testing. Analyzing small samples of blood, hair, or semen. 3. Research. Cloning genes. DNA sequencing. Studying gene expression, RT-PCR, qPCR. Site-directed mutagenesis. 4. Agriculture and environment. Identifying genetically modified organisms, GMOs. Detecting plant pathogens. Studying biodiversity and endangered species, DNA barcoding. 5. Biotechnology and industry. Producing recombinant DNA. Synthetic biology applications. Monitoring microbial contamination. Variants of PCR. To expand its uses, many specialized versions of PCR have been developed. RT-PCR, reverse transcription PCR, used for RNA, Example: detecting RNA viruses. qPCR, quantitative real-time PCR, measures DNA amplification in real time, used in COVID-19 testing. Multiplex PCR, amplifies multiple targets simultaneously. Nested PCR, increases specificity by using 2 sets of primers. Digital PCR, highly precise quantification of DNA. PCR is a powerful technique invented by Kary Mullis in 1983 that amplifies DNA in vitro. It requires template DNA, primers, TUC polymerase, dNTPS, buffer, and Mg superscript 2. It works in cycles of denaturation, annealing, and extension, and is widely applied in medicine, forensics, agriculture, and biotechnology. Thanks for watching.